Welcome to our webinar on Genomic Integrity G-Band Karyotyping for cell and gene therapy drug products. I'm Stephanie Elena, Vice President of Marketing at KromaTiD. Today, we're privileged to have Greg Hussar, Director of Logistics Operations, and Mike Vernich, Director of Service Operations, as our presenters. Before we begin, a quick note. If you have any questions during the webinar, please use the Q&A box. We'll address all questions at the conclusion of the session. Additionally, we'll be conducting a brief 5-minute survey afterward and would greatly appreciate your feedback. Thank you again for joining us today. Now, without further ado, I'll turn it over to Greg.
Good morning. My name is Greg, and Mike and I appreciate your choice to spend time with us this morning. This presentation is about our Genomic Integrity G-Band format, how it came about, its value to our customers in the gene and cell therapy space. So first, we're going to walk through a little bit of history and information. It's a staining method yielding dark and light bands. It was invented in 1902 by Gustav Giemsa. Again, it is a DNA stain, so AT-rich regions stain darker than GC-rich regions, and yields a direct visualization method. So you can see that example in the image to the right. The International System for Human Cytogenetic Nomenclature, the ISCN, provides an internationally agreed upon system for describing the banding pattern of chromosomes, which means the data from one lab translates to the data from another lab or from one institution to another. Single cell data, it's not like sequencing, which uses pooled DNA. The analyzed cells are viable immediately prior to fixation, which means it's a snapshot in time of those cells, so your sample. It's microscopic detection, so greater than 5 megabases, again, not submicroscopic. So G-banding detects a wide range of microscopic chromosomal abnormalities greater than 5 megabases. This includes additions, aneuploidies, deletions, duplications, inversions, and both balanced and unbalanced translocations. The assay also excels in identifying subclonal populations and/or mosaicism, so where variant is present in a small population of the analyzed cells. Applications of this in the gene and cell therapy space are genomic integrity assessment, so understanding the integrity and stability of a genome over time or across different experimental conditions. So for pre and post treatment evaluations, this could apply to a research batch of donor to an edited cell. It could be in a different stage of your process, so an engineering batch, for example, or even final drug product. Process-specific evaluations, so you can assess these genomic changes for things like payload delivery method comparisons, editing enzyme comparisons, number of rest days post-edit, even culture stress versus process stress. It's good for IPS clonality assessment, so looking at clonal status of induced pluripotent stem cells, and longitudinal studies, so looking at changes over time, so checking culture every 5 or 6 passages, for example. So we've been asked, what specific guidance has the FDA given to us? And we actually have not had any specific direct provided guidance from the FDA, only through our customers and the information that they get and request from us. But this webinar, which was given recently by the FDA for human gene therapy products, had the following question at the 45-minute mark in this presentation. Is G-band karyotyping sufficient for the assurance of chromosomal stability for a genome-edited gene therapy product? The FDA's response, it goes over several minutes, but basically says G-banding is not very sensitive and is also not a direct measure of the genomic DNA, but rather the secondary chromosomal structure. That said, within its limits, it is a workable measurement for many cell and gene therapy purposes if applied correctly, and many of our customers are being asked for this data. Also to note, our genomic integrity data has been used in multiple IND filings and is currently being used in multiple release tests for phase 1 clinical trials. We have GLP level processes and reporting for this, and we are working to establish GMP levels. So instability in drug product batches is detectable using G-banding. However, there are 3 factors that can limit the sensitivity of G-banding for early markers of instability. Resolution, again, G-banding has a resolution that varies by type, size, and location of structural variant. So 5 to 15 megabases is an estimated resolution, but there are variants that do not disrupt band patterns. Cell number. Typical G-banding cell counts of 20 cells with full analysis of only a subset of those cells makes reliable detection of low frequency events impossible. And lack of a direct comparator. Unstable genomes have similar hallmarks to rearranged but stable genomes, and so to truly assess the stability of a genome, a reference sample is required. So KromaTiD uses G-banding as the basis of a release test and has defined the genomic integrity specification to address some of these limitations. GI G-banding is done on pairs of samples to measure variance, and typically a full analysis performed on at least 100 cells to catch low frequency events that may be early markers of instability or developing subclonal population. This slide is about why standard karyotyping doesn't address the reporting needs for the gene and cell therapy world. So clinical G-band karyotyping was developed on patient samples, unaltered, and analyzed within days of collection. So a karyotype reasonably describes the chromosome status of the patient sample. But genomic integrity analysis on therapeutic batches is applied to cells that are isolated, expanded, treated, sorted, and cryopreserved. So each cell within that sample actually has its own sort of life experience, different cell-to-cell impacts of the treatment process, selection pressure during expansion, impact of repeated activation, stimulation.
So the way clinical cytogenetics reports abnormalities is based solely on the presence of a clonal activity in a given sample. So when we're dealing with the cell and gene therapy cell lines, this isn't necessarily the case. We're more looking for trends involving the customer's target edit sites rather than repeat observations of the same clones you would typically see in a cancer cell line. This next slide, we're going to go into more depth of why we decided to transition to more easier report for our customers to interpret. These are some of the questions that they typically had for us. And we're going to go look at the report on the left. Getting into higher cell counts, the pros are you're getting a more in-depth analysis. Detection limits are higher. However, you're going to get some random background noise and random loss aneuploidy detection that's going to pop up in your final karyotype, which is pretty typical when you're looking at 200 cells. The one on the left is a 100 cell report, and it's a normal female karyotype according to conventional cytogenetics. But as you can see, several abnormalities present in this case where you have 7q and 14q being involved in many abnormalities. This isn't necessary clonal activity, but it could be misrepair or could be a sign of genomic instability across the sample.
All right, so continuing on, this is a pretty busy slide. This is the poster that we presented at ASGCT. So we don't need to go through the whole thing. I'm going to focus in on a couple of points here to continue on from where Mike was. So this is looking at the reference sample and a treated sample in a pair and individual karyotype reports using standard or old style format. When you look at this, how do you get to a comparison? What is the meaningful comparison to make here for you as the client? And then how are you going to present this data to either your collaborators or to a regulated body? So if we move over to our parsed out format for genomic integrity, what we've changed over to is the ability to break out both categories and specific individual events. And we do that in a frequency-based report, both for the reference sample and the treated sample. And then that allows you to make a statistical comparison in the comparison report, and you get a look at ploidy, categorical variant comparison, and then a statistical comparison using Fisher's exact test to see if anything stands out as significantly different. And it then funnels down to a specific variant comparison as well. And again, you will see if there is something that is statistically relevant or not. And then again, we have a link to this included at the end of this, so you'll be able to take a look at this on your own. So don't worry if you think that I blasted past anything here. And that moves us on to this next aspect, which falls out of the comparison report. So statistically relevant, at what level do you want to or need to see this information? So since we are breaking it down to a specific event comparison level, you can use these numbers around confidence intervals to decide on, with other data that you have, to decide on a level of analysis. So for example, probably our most common requested amount at 100 cells puts you between 2 and 5% at a 95% confidence interval. And then 200 cells puts you between 1 to 2% at a 95% confidence interval. So this allows you to determine, along again with your other data and any requests by collaborators or regulated bodies, to establish a level of analysis.
So now I'm just gonna highlight one of our other assays. It's our screen karyotyping. It's a much higher resolution assay. We're talking in the kilobytes here, whereas G-banding is in the megabases. This isn't necessarily to say one is better than the other. It's more to highlight how complementary these two, when paired together, how powerful they are at detecting and placing chromosomes in the correct places. So the advantages of our screen assay, you'll see a very small inversion on chromosome 8p. This is actually one of our control cell lines. And if you look at the G-band picture here, it's not detectable, even at a very high resolution, which this ideogram is at 850. You're not able to see something like that. So that is one huge advantage, and it's pretty cool to see in this example. Other ones involve cryptic reciprocal translocations. You see the 4p and 11p being involved. That would be pretty difficult to detect in a G-band assay. So these are some of the advantages over G-banding. And inversely, here are some of the advantages G-band analysis has over screen. You'll see in the bottom left picture, here's an example of the picture above in the G-band. The third chromosome is obviously a ISO 7P. In the screen assay, you can't readily see this. It looks like it may be deleted, but you don't know. But thanks to the distinct banding patterns of G-bands, you're able to detect this, as well as the fourth chromosome being involved in a unbalanced translocation involving 7P and 11Q. Looks very similar in the screen assay to the one on the left. So unbalanced translocations, isochromosomes, smaller deletions, these are all things that G-band analysis is great at detecting and may not be able to see in the screen assay. But like I said before, this is a great way of analyzing a complex cell line concurrently, because you get whole genome single cell analysis and you can visualize what's going on, and they're very complementary of each other. And pairing these together, very awesome and cool to work with. And at the same time, you're able to confirm or deny abnormalities that you would catch in one that you wouldn't in the other.
And that concludes our presentation on this topic at this point, and we'll move us on to the question and answer portion of today's talk. And so again, we appreciate your time and are ready for any questions you might have. Looking at, sorry, trying to going to expand something here in my view, sorry about that, so we can see these as they come through. So Alexandros asks, do you provide service for mouse cells? We are able to do analysis for other assays, for hybridization-based assays on different species, as long as there is a reference genome that you want to test against. We do not specifically do G-band karyotyping for mouse cells. And then we have one saying, will targeting a higher number of cells make it more likely that smaller and smaller rearrangements will be detected. And basically, analyzing more cells won't change the resolution of the events that are detected, only the prevalence and the confidence interval around detecting lower prevalence events that would be detectable in that range, if that makes sense. Somebody's asking if we perform OGM assay. Can you?
Not familiar with that.
Can you expand on what you mean by that specific acronym, please? Oh, optical genome mapping. Sorry. Thank you. We do not do optical genome mapping, but our version of that again is screen, right? That's a whole genome assay that's based on direct hybridization, so sequence-based visualization, and it is also single cell data. Again, for karyotyping, specifically G-banding, human specific G-banding is where our expertise is based. If there was something specific in another species and there was a reference and a very specific goal, it would be a development type project, but not something that we would do standard. Sorry, the questions keep scrolling. So we have a question that's asking, to what extent is the report customizable to accommodate regulatory submission needs? Again, this is a standard. So if you think about the genomic integrity reports that we have, we've created a standard format, so we can supply that to our customer base. But when it comes to individual reporting needs, we're happy to work with our customers because it seems that, they all have their own different internal quality programs that require a specific language, specific, words to be either there or not. And we're happy to work on customizing our reports. Depending on the magnitude of the customization, there can be some charges involved with that. We've also been asked if if a customer wanted us to perform both G-banding analysis and either some form of FISH or DGH, another direct hybridization visualization method on the same samples, do we basically, how do we avoid bias in that analysis? So for example, would we look at the G-band analysis first and then go look at the outcome of the FISH analysis or look for specific events? And the answer to that actually would be, it sort of depends on the way the project is set up with the customer in advance. So for example, we can even do fully blinded analysis. You don't have to tell us which is your treated sample, which is your edited sample, until we get to the point where you actually want a statistical comparison, and then there needs to be an unblinding. But we can give you all of the data up front for an individual genomic reports, and then unblind it after that and apply the analysis. So we're happy to be tested in that fashion. But we've also had very specific requests from customers where they say, we have this data from another platform, another analysis platform, and we want you to look specifically for these events. So we can do guided as well. We've been asked about different levels of quality for the analysis that we provide. And currently, we have RUO levels. We also have GLP level of analysis. And again, we're working towards a GMP version of it, but that is not currently what we have. So we can do individual samples under a GLP level. We can do also full study programs to qualify both our method and a specific even release test for a customer for a specific cell line or product.
Screen versus G-band.
There's a question about which method is better and FDA accepted. If you're asking about screen or FISH or direct hybridization methods, which are sequence-based, versus G-banding, which is staining and area-based for the genome. We don't, again, have any specific FDA guidance for that, and no direct FDA interaction. So again, the interaction, the information we get is, for example, from that webinar, and then specifically from each of our customers that tells us the information that they need. So we sort of gather information as we go for what the FDA is looking for. We've been asked about what kind of support can we provide for merging this kind of data output if somebody already has data like sequencing or optical genome mapping. Again, we're happy to customize all of our data outputs with the understanding that depending on the magnitude of that need, there will be some cost associated with it. But our goal is to get our customers the data that they need for their samples for their use case. And that really is one of the primary driving goals for how we set up all of our projects and customer interactions. Because that is our product, right? The data and the process that we use to deliver that to our customers. A quiet group today. I expected some more questions. You know, I guess one of the things that I will say is we get lots of different questions from our customers, and quite often some of the questions are what's wrong with my sample, or is my sample okay, or you've seen lots of samples from lots of different companies, how does mine compare? And we will often get asked that same question, maybe 4 or 5 different worded versions within the same data review meeting. So again, that was one of the other drivers that moved us towards this alternate reporting format, which says like a fully prevalence-based, non-judgmental based, right? So even if something is significantly different between the comparator sample, the reference sample, and the treated sample, we don't place a value judgment on that. It's not good or bad. It's simply significantly different. And then you then, as the user, as the customer that has the process and the sample, will use your other data, anything else that you have, guidance from collaborators or the regulated body, to make an assessment based on that information, and you have statistical power to present. Anything else that we have? We've been asked about the amount of sample we need to, especially if somebody wanted to do both G-banding and screen in a side-by-side experiment.
That's going to depend on how many cells analyzed you want for each assay.
Yep. So there are some amounts. Also, typically we would prefer to have a separate prep for G-banding and for screen, which is a DGH based method. although we can do the analysis on the same sample. But so typically 1 million cells in fixed process at the end would give us enough for say 20 a 20 cell analysis in G-band and a 20 to 50 cell analysis in screen if the quality is good. If you want to do higher counts, so for example, if somebody wants 200 cells of analysis, we would prefer to have about 200 cells of G-band. We'd prefer to have about 5 million cells in a fixed process at a good quality, and then 50 cells of screen on top of that would probably be, you know, another 1 million to 2 million cells. And 50 is a standard offering, a standard amount of cells that we would provide an analysis for screen. You can do more as well or less, but 50 is a pretty good picture. All right, we seem to have come to the end of questions. So once again, I will say, I'm just gonna scroll down for a minute. This is an example of some of the customer questions. And then we're going to provide this slide that you'll get along with this at the end. So it has links to our web page for GI and genomic integrity, as well as report links, and then a link to the poster that was presented at ASGCT for this. Anything you want to inquire about can be inquired at sales@KromaTiD.com. So again, thank you for your time today, and hopefully we will get to work with some of you in the near future.